overall goal of this protocol is to increase the quantity of isolated double-stranded DNA and subsequent high-throughput sequencing libraries from herbarium specimens, maximizing their utility in phylogenomic studies.
This method unlocks the potential of herbarium specimens that would otherwise not be useful for sequencing projects as genomic sequences because of extensive sample degradation. The main advantage of this technique is that it is robust enough that rare and historically significant herbarium specimens can be used without the need for extensive destructive sampling.
This technique extends the depth of taxon sampling in systematics-based projects. Species acquisition is a limiting step, but with this protocol, historical collections can be easily used into these studies.
Add liquid nitrogen and 30 to 50 milligrams of sterilized sand to grind 1 centimeter square of pre-selected herbarium tissue to fine powder in pre-chilled mortar and pestle.
Adequate grinding is essential for successful DNA isolation, and care should be taken that the sample is completely pulverized.
Next, add the frozen powder to two 2-milliliter tubes, filling no more than half of the tube volume. Then, add 600 microliters of warm CTAB solution to each tube. Thoroughly mix the powder by inverting and vortexing. Then, incubate the samples in a water bath maintained at 65 degrees Celsius for 1 to 1.5 hours. In the meantime, vortex every 15 minutes. Then, centrifuge the samples at